Welcome to today's video. Calculations are the backbone of successful cell culture experiments. Whether it's preparing media, seeding cells, or scaling up cultures, getting the math right ensures consistent results. In this video, we'll simplify these calculations, provide examples, and finish with practice problems to boost your confidence. Let's start with the basics, calculating cell concentration and viability. These two go hand in hand when working with a hemocytometer and trypan blue staining. First, to calculate cell concentration, use this formula. Cell concentration equals the average number of cells counted per square, multiplied by the dilution factor, multiplied by 10 to the power of 4. This gives you the total number of cells in 1 milliliter of suspension. Easy, right? Let's try an example. Say you counted an average of 50 cells per square, and you diluted your sample 1 to 2. Plugging into the formula, 50 times 2 times 10 to the power of 4 equals 1 million cells per milliliter. Pretty straightforward. Now let's talk about viability. To calculate cell viability as a percentage, here's what you do. Divide the number of live cells by the total number of cells counted, then multiply by 100. For example, if you counted 120 live cells and 30 dead cells, that's a total of 150 cells. So, 120 divided by 150 multiplied by 100 gives you 80% viability. Not bad. Next up, let's talk about seeding density. Seeding density is basically how many cells you want to plate in a specific area or volume. Don't worry, it's easier than it sounds. Here's the formula you need. Volume to seed in milliliters equals the desired cell number divided by the cell concentration. Okay, let's try an example. Say you want 1 million cells per well, and your cell suspension has a concentration of 2 million cells per milliliter. Using the formula, you divide 1 million by 2 million. That gives you 0.5 milliliters. So, you'd need to pipette 0.5 milliliters of your cell suspension into the well. Remember, always double-check your math to make sure you're plating the right number of cells. Oh, and one more thing. When you're seeding cells, make sure to mix your cell suspension well, so every pipette pull has the same concentration. Now, let's move on to media preparation, an essential part of any cell culture experiment. Don't worry, the math here is pretty simple. You just need this formula. The concentration of the stock solution multiplied by its volume equals the concentration of the final solution multiplied by its volume, or, more simply, C1 times V1 equals C2 times V2. Let's break it down with an example. Say you need 500 milliliters of 10% FBS in your culture medium, but your FBS stock is 100%. To calculate how much FBS to add, divide the desired concentration multiplied by the desired volume by the stock concentration. So, that's 10 times 500 divided by 100. The answer is 50 milliliters of FBS. Now, subtract that 50 milliliters from the total volume of 500 milliliters. You'll need to add 450 milliliters of your basal medium to make the final solution. Make sense? Excellent. Let's move on. Now, let's dive into scaling up cultures. This comes in handy when you need more cells for an experiment or a larger batch. Don't worry, it's not complicated. Here's the formula. Volume to seed equals the desired cell number divided by the cell concentration. Here's an example. Say you're transferring to a 175 square centimeter flask, and you need 5 million cells. Your cell suspension has a concentration of 1 million cells per milliliter. Just divide 5 million by 1 million. That's 5 milliliters of cell suspension. Finally, let's talk about cell dissociation and subculturing, a routine yet essential part of cell culture. When your cells grow too crowded, you'll need to split them into new flasks. Here's a handy formula for split ratios. Split ratio equals the volume of the old culture divided by the total volume after splitting. Let's go through an example. If you have 10 milliliters of cells in your flask and you want to split them 1 to 5, simply multiply your initial volume by 5. This gives you 50 milliliters. So, you'll add 40 milliliters of fresh medium to your 10 milliliters of cells. That's it. You've split your cells 1 to 5. See how that works? Now, it's your turn. Let's try some practice problems together. 1. You counted 80 cells per square on average with a 1 to 2 dilution. What's the cell concentration in cells per milliliter? 2. You're preparing 250 milliliters of 10% FBS from a 100% stock. How much FBS and medium do you need to mix? 3. If you have 15 milliliters of cell suspension and want to split it 1:4, what's the total volume and how much fresh medium do you need to add? Take a moment to solve these. Feel free to pause the video if needed. When you're ready, let us know your answers in the comments section. And that's a wrap. Today, we covered essential calculations for cell culture, including cell concentration, seeding density, media preparation, scaling up, and subculturing. Mastering these will elevate your lab skills. If you found this helpful, like and subscribe for more lab tutorials. Thanks for watching and happy culturing!